Hi, everyone. Thank you again for joining us today for Dana-Farber's Targeted Protein Degradation webinar series. After a quick summer break, we're really excited to have Stuart Schreiber as our opening speaker. So Professor Schreiber is the Morris Loeb Professor of Chemistry and Chemical Biology at Harvard University, a co-founder of the Broad Institute of Harvard and MIT, and has been a member of the National Academy of Sciences since 1995. Stuart completed his graduate studies at Harvard University under the supervision of R.B. Woodward and Yoshi Kishi, after which he joined the faculty at Yale University in 1981. In 1988, he returned to Harvard, where he has been ever since. His research has been recognized through numerous awards, most recently the Wolf Prize in Chemistry. And in addition to an exceptional research career, Professor Schreiber has founded and served on the board of numerous biotech companies, beginning with Vertex Pharmaceuticals. And these companies have since developed many novel therapeutics that are FDA-approved drugs or are currently in clinical trials. As Stuart highlighted in his recent review, 2021 marks the 30th anniversary of the revelation that cyclosporin A and FK506 act by a novel mechanism, which he coined molecular glues. And this discovery led conceptually to the targeted degradation of proteins by small molecule PROTACs. And so it is only fitting that we have Stuart kick off the second year of this webinar series. And with that, I'll hand it over to you, Stuart.
Thank you, Alyssa. And good afternoon, everyone. It's a real pleasure to be here, part of this targeted protein degradation series. I'm excited about your targeted protein degradation series. I confess I didn't know a lot about it until this weekend when, trying to think about what to share with you. I went online and I went to your YouTube channel and I'm absolutely blown away. I will tell you that I only had a chance to sort of spot check some of the talks, but I've decided that I want to set aside real quality time to see a number of the presenters that have preceded me, and I feel like just pedagogically it's a gold mine. So delighted to be able to be part of this, and hope that what I share is the kind of high quality that I saw in some of the spot checked presentations. Now, first I want to say that you can already tell maybe from the title, I'm not actually going to talk about targeted protein degradation per se. But on the other hand, as I'm looking at this little image, if you look down in panel C, there's Jay Bradner's Dtag system. There's the familiar thalidomide lenalidomide system. There's our PROTAC system. So, of course, targeted protein degradation through induced proximity is a subset of the world of molecular glues. One of the things I want to share with you, and I will touch on the targeted protein degradation because I think it's really obviously the most powerful demonstration thus far of of molecules that induce protein-protein associations. But I think the field is in its growth phase, and I'm going to try to highlight lots of other things you can do with these kinds of molecules, molecular glues, and hopefully that'll fit nicely into the seminar series. As Alyssa mentioned, for those of you who'd like to learn a little bit more than what I'm going to share, I did write a perspective that appeared in the first issue of this year's edition of Cell called The Rise of Molecular Glues. So let's jump right in. Although, give me just a second, I just want to look at my timing. Hopefully stay on time. Okay, so you were back to the presentation. Let's begin with the basics. So when small molecules bind dynamic proteins in cells, they change their biophysical properties. And we can think, I'd like to share with you the idea that we can think of the small molecule protein complex as a neoprotein. One of the more, turns out, common things that happens upon binding is that the compound alters the interactome of its target. inducing protein associations. And the consequence of this, the consequences are many. The binding can and altered interactome can change the lifetime of the protein. And small molecules can serve as stabilizers. It can decrease the lifetime of the protein. Compounds can be destabilizers or even degraders. But lots of other things can happen. So in the case of the Vertex cystic fibrosis drugs, compound binds its target, changes its interactome, and causes its target to translocate to a different part of the body within a cell. So cellular localization can be affected by these binding events. I'm going to end today by sharing with you a new direction that my lab is focused on, a speculative new direction that I hope you'll find of interest. Okay, one other point I wanted to make, because we're in a chemical biology audience here. I'll bet you that many of you, like myself, have been asked many times, what are the differences between genetic perturbations and chemical perturbations? And we know the answer is that there are many differences. What it took me a long time to realize is that the nature of that question, I think, most often reflects the idea that genetic perturbations are laboratory perturbations, like knockouts and knockdowns. And that is where then the differences are really substantial. But what I've come to think, and I'd like to share with you, is that actually where small molecule perturbations and genetic perturbations align very frequently are natural missense mutations under natural selection. So, for example, in BRAF, the somatic alteration of a valine into a glutamic acid, the VDE mutation, changes the interactome. Haiyan Fu's lab has shown this very beautifully, inducing a neoprotein interaction that is at the heart of the oncogenic properties of this allele of BRAF, and it involves induced protein association. So I think, and it kind of makes sense, right? Changing an amino acid side chain in a hotspot of a protein from a valine to a glutamic acid is kind of like a small molecule binding in that site. So altered interactomes. Okay. Now, this feature of compounds, as Alyssa mentioned, the ability to induce protein associations or functions, molecular glues, was first illuminated in the study of the mechanism of action of these two natural products, cyclosporin and FK506. This was work that was led by a then postdoc in the lab, Jun Liu, and I'm going to mention some of Jun's work in his independent lab at Hopkins in a minute. Jun, using largely affinity chromatography, discovered that these two compounds target the same protein, the calcium-dependent protein phosphatase calcineurin, or PP2B. Now, he showed that the way they occur is unusual in two respects. One, point to FK506 for a moment. Rather than binding in the enzyme active site where the asterisk is, these compounds block the entry portal for a subset of the normal substrates. So they're substrate selective inhibitors, in this case of a phosphatase. Now, two, the way they do so, as you can tell, is a molecular glue. And in the case of FK506 and another natural product I'm going to mention in a moment called Rapamycin, both bind a protein that was also discovered with affinity chromatography that we named FK506 and rapamycin binding protein of molecular weight 12,000, which we shortened to FKBP12. FK506 binds FKBP12 with very high affinity, a dissociation constant of 200 picomolar. However, neither FK506 nor FKBP12 have appreciable interactions with calcineurin. There's some debate about this, but at best it's tens of micromolar. Yet this small molecule protein complex binds cooperatively. The binding of the complex involves calcineurin interacting with FK506 and FKBP12, and therein lies the cooperativity. And so now you have a 1 nanomolar binding constant of the ternary complex. And exactly the same story emerged, as Jun showed, with cyclosporin. Although its presenter protein, a term I'll be using, is cyclophilin, also found by affinity chromatography. But now the cyclophilin-cyclosporin complex cooperatively binds calcineurin. Now, when this work was underway in my lab, we were collaborating with Don Wiley. And Don Wiley's lab had just discovered and determined the crystal structure of the first MHC protein. You all remember the famous extra electron density in the MHC, which he surmised was an antigenic peptide that had come along in the co-crystallization. And of course, we now know the MHC protein can bind different antigenic peptides, and depending on which peptide is present will determine which T cell receptor is engaged. This seemed really similar. And so I wrote a paper in the next year in Cell where I drew analogy to these two systems and coined the term molecular glue. So the molecular glue was not only in the context of the two natural products, but was in the context of MHC peptide interactions interacting with T cell receptors. Now, around this time, I have to say, I thought this was an amazing, feat of natural products, but we chemists would never be able to do this. It was only a billion years of natural selection that would allow this kind of ternary complex to form. Well, that idea changed about 10 years later when then graduate student Steve Haggerty was working in the lab and discovered this simple synthetic compound we called synthetic stabilizer A, or SynStab A. And Steve showed that this compound, both in vitro and in cells, induces association of alpha and beta tubulin to stabilize microtubules in vitro and in cells. In exactly the same way the very complex natural products, Taxol, discodermalide, apotholone does. Yet it was a simple off-the-shelf chemical. And these days, we now recognize just how common this phenomenon is. So many times in the lab, when we have a tool compound, we know its target, we'll do a chemoproteomic pull down with and without the compound. And more often than not, we see changes in the interactome, often stoichiometric changes that often involve proteins of real relevance to the mechanism of action. So this, I'm beginning to believe, is more common than we'd ever imagined. I mentioned rapamycin. We did know at this exact same time, we knew rapamycin was also a molecular glue. We'd done experiments to more or less prove that, but the identity of its partner was parked for a couple of years because we were so excited about using FK506 and cyclosporin to map out the signal transduction pathway, the calcium calcineurin NFAT pathway. NFAT had just been discovered by Jerry Crabtree, and we joined forces with the Jerry Crabtree lab to map out this membrane to nucleus signaling pathway. So a couple of years later, then graduate student Eric Brown in my lab decided to tackle this rapamycin problem. And he used almost exactly the same procedure that Jun had used, affinity chromatography, and out comes the target right straight away, the mTOR protein. Now, it turns out at the same time, then graduate student David Sabatini had conceived of this same problem and working independently in his lab, and Saul Snyder's lab as a graduate student, did the same kind of affinity chromatography experiments and also found the same protein, a previously unknown protein kinase, which is now called mTOR. mTOR stands for mechanistic target of rapamycin, because the target of rapamycin, of course, is FKBP12. Remember, super high affinity, cooperative binding then to mTOR. That's how mTOR was discovered. I have to say that over the years, David Sabatini and his lab have just done remarkable, groundbreaking experiments to look way beyond this into the interactome of mTOR, the discovery of mTORC complexes, and really truly illuminate the nutrient response signaling pathway. It's been a lot of fun to watch the Sabatini lab advance this area. Now, rapamycin blocks the entry portal, familiar theme again, doesn't bind to the ATP binding site of the kinase, and it selectively inhibits certain substrates, in fact, of certain mTORC complexes, a familiar theme. Some of you in the audience will be surprised to hear that at this point in time, in the late 1980s, phosphatases and protein phosphatase, protein kinases had been declared as undruggable. In the case of kinases, this is because the ATP binding pocket was so conserved. So, well, first of all, Sandimmune, cyclosporin, was an approved drug. It was a blockbuster drug. So that, removed the myth of phosphatases being undruggable. And just a couple of years later, rapamycin became approved as the first protein kinase inhibitor called sirolimus, proving that you could target kinases. And of course, we now know that protein kinase is one of the most common classes of targets for drugs. However, I think it's interesting that the first kinase inhibitor worked by this molecular glue mechanism by binding to what David Liu would call an exosite and having substrate selectivity on the kinase activity. And I think what I hope at the end you'll see, the new ability to pre-select targets, presenters and targets for discovery of molecular glues means it's, in my opinion, it's time to go back and really re-examine lots of protein kinase targets and find this rapamycin mechanism, molecular glue mechanism, because there are some really, there are a number of features of this mechanism that I think can be very useful as medicines. Recently, WDB002, a third natural product, was reported that also binds FKBP12, also acts as a molecular glue, and its target is a different one. So like those MHC peptide interactions, you now have a third natural product. In this case, binding cooperatively as the complex, as the crystal structure shows, this compound targets the coiled coil region of CEP250. Could you imagine finding a small molecule that all by itself binds with high potency and specificity to a very specific region of an otherwise featureless coiled coil? I think that would be difficult, and yet through the cooperative interactions of the protein with the small molecule and the presenter, this can happen. Now, so that's 3 natural products with FKBP12. Well, Jun Liu recently extended this in a very dramatic way. So a little back up, 30 years ago, my lab also synthesized this compound called 506BD. What we did is we carved out the linker or connector in rapamycin and FK506, and we replaced it with this alternative one. This one does not bind calcineurin, and it does not bind mTOR. In fact, we used this compound and showed that it was an antagonist of both compounds, binding competitively to the presenter. And that's really what got us onto the whole gain of function molecular glue idea. Now, what Jun did recently in his lab at Johns Hopkins is he replaced this linker with 45,000 dipeptidoid-like molecules, made them individually, arrayed them, and then used these in phenotypic screens. And he finds really novel activities over and over that are FKBP12 dependent, meaning they're molecular glues. Like Rapidocin, he reported in this paper last year, which binds the transporter, SLC29A1, in an FKBP12 dependent manner as a molecular glue. So FKBP12 alone can be used and repurposed over and over. But of course, it's not just FKBP12. It's all kinds of presenter proteins. So what I'm going to do is, you all know this, molecular glue is being discovered, weekly, really dramatic cases. I just picked a few of some of my favorites to try to highlight the range of activities that small molecules have in their mechanism of action, the kinds of things that would have been, I think, very difficult to imagine in the absence of the molecular glue concept. Angela Kohler at MIT, a champion of using chemical biology to target transcription factors and going after the toughest one, MYC. Well, she recognized that MYC is stabilized by the max heterodimer, that they're in equilibrium. And she discovered this diversity-oriented synthesis derived MYC degrader. This compound binds MAX, pulls it out of the complex, and stabilizes the MAX-MAX homodimer. A novel way to make targeted protein degradation, in this case of MYC. Pei Zhao was interested in POL-Z, the polymerase. POL-Z forms an obligate interaction with Rev1, Rev7, but they're in equilibrium. And he found this compound that pulls Rev1 out of the complex and stabilizes a Rev1, Rev1 homodimeric structure as a molecular glue and inhibits the polymerase. In a minute, I'm going to tell you about a kind of workaround that we came up with to overcome the perception back then that you need to use natural products. Because what we were really interested in doing was not just creating neoproteins and neoprotein-protein interactions, but we really wanted to be able to create neosubstrate interactions with enzymes. I'll show you how we did that in a moment. But to come forward to the molecular glue story, as everybody here knows, we now know lenalidomide and thalidomide are molecular glues that do exactly this. They create neosubstrates by, in this case, binding to cereblon. Cereblon, of course, is a multimeric protein complex having E3 ligase activity, and the neosubstrate could be Icaros, could be CK1 alpha, shown here, now become ubiquitinated. Chemistry occurs with a neosubstrate. Lots of labs contributed to this. Bill Kalin, Jay Bradner, at least Jay is a previous speaker. My own lab, the privilege of working with Steve Carr and Ben Ebert in illuminating, and I'm neglecting a number of other labs that made major contributions. Now, molecular glues, last point, can act intramolecularly. Here's a compound that Derek Taylor and Gautam Narla discovered that affect the equilibrium of interaction subunits in a different phosphatase, not PP2B, but PP2A. Very reminiscent of FK506 with calcineurin and rapamycin with mTOR. And one of my favorites comes from Kong Liu and Zhidong Zhu at Novartis in Basel. These workers were also looking at a protein phosphatase, this one a tyrosine protein phosphatase, SHP2. SHP2 is, I'm sure everybody here knows, is a signaling protein that is auto-inhibited by its SH2 domain interacting in the enzyme active site until mitogenic signaling comes along, creating phosphatyrosines that compete with the SH2, pry it open up the enzyme active site. However, during oncogenesis, oncogenic alleles of SHP2 are now known that disrupt this interaction, leaving the protein constitutively active. So Kong Liu and Zhidong Zhu and colleagues at Novartis discovered amazingly an intramolecular molecular glue that pulls this intramolecular interaction back together, even in the oncogenic case, thereby inhibiting it. And then the last point, they showed that the active form of the protein enters into a liquid-liquid phase separated condensate. And they showed that when you add the intramolecular glue, you pull the protein out of the condensate. I give you this example because, you know, we're often, again, as chemical biologists, our colleagues will say, like, find the target of my compound. And I always say, that's just one step along the way. Like really it's molecular mechanism of action, because who could have imagined that we would have compounds that work on selective targets by pulling them out of liquid condensates as their true mechanism of action. So a year ago, then graduate student Chris Gary and I wrote a perspective in Nature Chemical Biology on the unifying principles of these bifunctional proximity-inducing small molecules. Here's my confession. The reason we did this in part is that there's been a proliferation of names given to these, and mea culpa, my lab is more guilty than anyone, because I told you we started calling these molecular glues, then we called them dimerizers, then we called them chemical inducers of proximity. Former postdoc Craig Cruz, I believe, is the one who popularized the term protax. And there's lots of others. Jay has used the term imids. The point of this paper was that the underlying, don't get confused by the different labels. The underlying principles are the same. small molecules can induce protein-protein associations, creating neoproteins, neo-interactions, and neo-substrates. And in this perspective, we pointed out that these compounds can be extremely complicated and extremely simple. They can be from nature and natural selection. They can come from the laboratory. In fact, the full matrix, every possibility. Okay, now let me return to my anchor figure. I told you, I used to call it natural product envy. I thought this would never be done with chemists couldn't do this kind of thing. So we had a workaround. We decided we could borrow these natural products and mix and match them to make all kinds of molecular glues having linkers. And so linker was the key. The linker, like in rapamycin that prevents rigidity, well, now got converted into this linker in FK1012. This was a compound synthesized in the lab by Stephen Diver, who was a postdoc. You'll note, and it exemplifies this idea of molecular glues with linkers. So he took FK506 in a single reaction, did an olefin metathesis, spit out ethylene, and joined together two FK506s. Now, this compound, or this substituent, I could call it, on FK506, is mutually exclusive with calcineurin binding. You prevent the calcineurin interaction. However, it allows the interaction with another FKBP. Then we got the idea of making genetic fusion proteins. We would fuse, for example, shown here, a myristylation sequence onto FKBP12, and we would carve out the cytoplasmic domain of the Fas receptor that normally gets activated by the Fas ligand by bringing two of the receptors together, and the weak proteolytic activity of the Fas receptor in intracellular tail would act on itself, cut itself off, and set off the death signal of Fas receptor signaling. So we made a transgenic mouse. We used a tissue-specific promoter, made these transgenes only in the thymocyte, and it was a completely uninteresting animal. Nothing happened, except this protein is made in thymocytes. However, when we treated it with FK1012, we could then induce the interaction, and with spatial and temporal control, we could turn on a signaling pathway, leading to cell degradation, cellular ablation of a specific CD4/CD8 double positive thymocyte. So through this notion of molecular glues with linkers. And this, by the way, now has been shown to work in humans. So Jerry Crabtree and I started a company called Area Gene Therapeutics, and that they partnered with Bellicum Pharmaceuticals. AGT made Remiducid, licensed it to Bellicum. And Bellicum recently in New England Journal of Medicine has reported that, to my knowledge, the first synthetic molecular glues having these linkers to be shown to be effective in humans. So what they did is prior to bone marrow transplantations, they ran a study with leukemia patients that were undergoing bone marrow transplantation. Prior to transplantation, they transduced the purified hematopoietic stem cells from the donor with this very similar construct, myristolated FKBP Fas. 5 of the patients developed graft versus host disease, where the grafted new immune system started to attack different tissues in the body. They were able to cure these 5 patients with 1 single dose of Remiducid by turning on the signaling pathway and ablating the autoreactive T cells. Now, we also, through Ariad and Area Gene Therapeutics, made these kits freely available. Over 1,200 kits were made available to academic labs, and over 2,000 papers emerged showing over and over and over again the broad generality of molecular glues with linkers with these genetic fusion constructs to create neo-substrate interactions with enzymes and to turn on chemistry through cell surface receptors, lots of cellular processes. Jerry Crabtree's lab has been very active in looking at this tool to study gene expression through chromatin remodeling, chromatin methylation, gene expression, gene repression. It's been used for protein degradation, transport, moving membrane localization, a very robust system. However, it always involved using genetic fusions. So lots of very exciting time in the lab, lots of group members brainstorming about all kinds of ways to get around the need of genetic fusions. So I'm skipping over lots of steps along the way. I just again want to highlight what I view as some of for me, the big conceptual breakthroughs that really had such an impact. So one is the work of Jay Bradner when he started up his lab at the medical school and collaborated with Nathaniel Gray. So they invented the Dtag system. I'm sure maybe Jay talked about this in his lecture, but just to remind you, this is one of the most powerful ways for tools for functional genomics today. And what they realized is that they could synthesize one single compound, the Dtag reagent, with the cereblon binding element linked to the FKBP12 binding. And then they could attach an FKBP to whatever target they want or family of targets that they want. And adding a small molecule, they could induce the targeted degradation through this proximal relationship. And again, I'm sure everybody here in this audience knows this. And then the next big breakthrough, of course, was to get rid of the need for any fusion protein. So this is what underlies, again, I don't need to tell this group, the PROTACs, the work of Craig Cruz and Ray Deshays, Jay Bradner, and many others now for targeted protein degradation, or PROTACs. It got extended to extracellular degradation. I think there was some news recently about LITACs. At least 3 labs I'm aware of, former group members David Spiegel, Weiping Tang, developing their version of LITACs, and of course, Carolyn Bertozzi developing her LITAC system for extracellular protein degradation. And then recently at Genentech, Yamazawa and Staben developed Forks. This was a compound that binds an adapter unit of a phosphatase linked to a binding element to a target protein and inducing protein dephosphorylation. And Amit Chowdhury making Fix by a molecule that binds C1 domains of protein kinases, and then linking it up to a binding element for a protein and inducing protein phosphorylation. So just so many new things that chemical biology is able to do by creating neo-substrate interactions in cells. All right, so for me now, as a major next step, more than 10 years ago, what this really did was make me think, rather than thinking about disease and chemical biology and medicine as like find an enzyme, make an enzyme inhibitor, see if it works in humans, why don't we just think about finding binders? And if we could find a binder, we could see, is it a molecular glue? Does it change the lifetime? Does it change the interactome? So a binder first, function later approach. That was what we wanted to take. And so to do so, we recognized sort of 2 challenges. 1, what are the compounds we should use? And 2, what are the assays that we should use? I'm going to highlight some of the thinking in both of these areas. So what compounds do we use? Well, we were really influenced by the work of Jim Wells and his concept of hotspots and proteins. These hotspots are obviously 3-dimensional topographies, and we had the ridiculously simple concept. If we just include a lot of sp3 content in candidate binders, even if that creates stereogenicity, well, we could exploit that by making every possible stereoisomer. Maybe these compounds would be well suited for binding. And I don't mean to overstress this, but candidate binders can come from commercial vendors. They don't look just like this, but I'm trying to exaggerate a point. These compounds tend to have a high sp2 content. Why? Because the chemistry to make such molecules is robust and been around a long, long time. But the organic chemists in the audience know that today modern asymmetric synthesis is no different from the classic chemistry. You can make molecules having 3-dimensional topographies just as easily, just as readily, same number of steps using modern asymmetric synthesis. So we set out to make 100,000 of these compounds, pure compounds. We put them in barcoded tubes. And we first explored whether this idea that different chemistry would yield different outcomes in the context of phenotypic screens. So fast forward 3 years ago, Chris Gary and I again wrote a perspective, Nature Review and Drug Discovery, because we had this great opportunity. My lab was part of the molecular library program. The NIH assigned us 350,000 of these commercial vendor compounds, hand-selected by a blue ribbon panel of medicinal chemists, chaired by Chris Lipinski. And then we asked, could we, in our assays, our screens that we were assigned, could we include our 100,000 compounds from what we call diversity-oriented synthesis? And they permitted us to do that. This gave us, we ran 160 phenotypic screens, and it gave us an opportunity to do side-by-side comparisons. That's the topic of this perspective, where we found over and over compounds having this high sp3 content had novel mechanism of actions and novel selectivity. So I'm not saying that it's better by any means, it's just different. And it's, you know, we really want, we want compound collections that have high sp2 content and high sp3 content. And that was the point as judged by phenotypic assays. But I just told you we want to run binding assays. What do we know about that? Not a lot. So we're trying to figure out whether this will make a difference in binding assays also. Now, what binding assays do we use? Well, I'll tell you, there are a number of binding assays out there, lots of really interesting things. My own bias was that the most exciting, and I thought maybe a productive one, would be the chemoproteomic methods that have been pioneered by Ben Cravat and now lots of other labs. But my lab doesn't use chemoproteomics, we're not familiar with it. So rather than going down that path, I picked up the phone, I called Ben Cravat and I said, hey, here's a proposal. Why don't we join forces? Why don't we join our labs together, which we did, and we meet monthly in a video linkage between our groups. We share all of our data for these binding first, function later experiments, where chemoproteomics comes from the Cravat lab. What did my lab do? So we said, my second favorite on the list would be to use the concept of DNA encoded libraries pioneered and conceptualized by Sidney Brenner, Richard Lerner, also 30 years ago. Now, of course, you all know lots of papers, lots of work. Honestly, though, I still think it's a field that has, we still have to learn a lot, because one of the reasons is that a lot of the Dell discovery work is done in contract research organizations like pharmaceutical companies that pay a lot of money. Your typical academic laboratory doesn't have routine access to this technology. Also, the chemistry requires concomitant organic synthesis and DNA ligation. DNA ligation is done in water and it yields DNA molecules, and that can be limiting in terms of the numbers of types of reactions you can perform. So what happened? I remembered that when Jay was a postdoc in my lab, he conceived of a new idea that he called do-si-do. He wanted to do diversity-oriented synthesis on encoded deoxynucleotides, and he wrote a grant and he got it funded. He was going to launch this whole area, but he also got a job offer from the Harvard Medical School, and he took that offer, and unfortunately the grant wasn't transferable. As we all know, Jay went on to become president of Novartis, and so like I called up Ben, I called up Jay and said, hey Jay, time to get the band back together. Why don't we collaborate? Because Novartis is a real expert in this Dell technology, especially Fred Burst in Basel, with whom we've collaborated. And we're so grateful to Fred because he's been so generous in teaching us how to do this. We also collaborated with Fred Zikri and Corinne Briner here in Cambridge. The three of us are the co-leads on this project, and always with the support of Jay Bradner. And so here's what we decided to do. Try to break through and make this more accessible. You know, Jay is a real advocate of open source science. So we agreed we're not going to file intellectual patents for intellectual property. We're going to make everything freely available, certainly the knowledge, and to the degree that resources allow, make the actual reagents available to academic labs. That's our goal. We're heading towards that. But we had a couple of science ideas. First, what if we try to innovate in new DNA compatible syntheses? So I'm going to show you how that happened. Matthias Westphal joined our team as a Novartis postdoctoral scholar. Matthias was trained with Eric Carrera, and he was excited by the work of Neil Garg, Chrisel Schlosser, and other labs on strain accelerated cycloaddition. So the idea that these reactions, like making a cyclic allene, maybe an allene, but it may be a distorted allene that has a whole lot of diradical character, an sp2, sp3 vicinal diradical. Maybe these are stable in water. They should be. Maybe they're compatible with DNA. They should be. And so Matthias called up Neil Garg. His lab has been very generous, giving us lots of protocols and so forth. But we ran these reactions in water on DNA. And Matthias was thrilled that cesium fluoride under DEL synthesis conditions generated in situ the strain cycloalene. And when he added a diazo carbomethylide, he could undergo cycloaddition. And this was not your garden variety. I mean, this was an amazing reaction on DNA in water. It's as good as reductive amination and acylation, some of the most robust reactions for DEL syntheses. Yet it's a stereoselective 2,3 cycloaddition yielding three-dimensional structures. Here's the LC trace of this. It's not cherry-picked. It's true of different hydrozids combined with different aldehydes in situ to undergo these cycloadditions. We could use different hydroxylamines in red with different aldehydes in green, in situ generating nitrones, in situ trapping the strain cycloalene to give these products. And according to the Garg protocol, but now on DNA, slightly electron deficient olefins undergo stereoselective 2+2 cycloadditions with the same efficiency and selectivity as I showed you in that LC trace. So this is exciting to us. We think 3 new postdocs, John Knapp, Mathieu Richter, and Benji Tang, are applying this basic chemistry to different instantiations of DELs, and we'll see where that's going to take us in the future. Now, the second approach, it was the J. Bradner do-si-do approach. Liam Hudson joined my group and our team as a Novartis postdoctoral scholar. And he said, Stuart, I'm walking around the lab and I'm seeing all these jars of all these intermediates for this 100,000 DOS library synthesis, 15-gram quantities of, he found 285 of these, winnowed down to the ones that were pure and the ones that had orthogonal functionality, like a protected amine, an aryl halide, and a hydroxymethyl group. And he performed a DEL synthesis. So I thought what I would do for some of you who may not be familiar with this split pool strategy of Brenner and Lerner, actually walk you through how Liam prepared his first DEL using this concept. I'll tell you that the hard part is to quality control building block coupling reactions and concomitant DNA tag ligations in water on DNA one at a time. That was the really hard step. An intermediate step is a little mini library where we make enough compounds that we could use LC mass spec to ask, are the 20, 25 compounds that we think we made all present and nothing else? And hopefully in roughly equal molar concentrations. All of that was done in advance. Now the DEL synthesis. So after picking 56 skeletons, wherever you see a sphere, that's a stereogenic element, and wherever there's a stereogenic element, Liam included every possible stereoisomer. So 4 of these, 8 of these. Now, split the 56 skeletons into separate flasks, attach a DNA headpiece to each. Next, ligate a forward primer and barcode, the first barcode. This barcode here tells us which skeleton was in that flask. We then pool them together, split into 600 separate reaction flasks, encode with a second barcode. And that barcode tells us which building block are we about next to add to that flask. Sulfonylation, reductive amination, acylation is performed, with pure reagents in these flasks. Now we pool those, separate them into 38 separate reaction flasks, encode the third barcode. This is going to tell us which building block we're going to add through Negishi transition metal coupling reactions on aryl iodides and aryl bromides. We then pool those together. We ligate the fourth barcode. This is the library barcode. This is a barcode that says this library was made by Liam Hudson on this day in this lab notebook, yielding this collection of compounds. this allows us to take the 10 or a dozen now different libraries, and if we wish, mix them together, read out the barcode, and remind us from which library did the compound come. And then we add a closing primer. So we make, Liam made over 3 million compounds with forward and reverse primers for PCR amplification. These are 55,000 molecular weight compounds with the ultimate tail wagging the dog, the little 400 to 500 molecular weight compounds sitting over here. And we hope that whatever binding activity we find is coming from this piece. Now, how do we know that? First, just on a glance, I hope you can see, I think you agree, these compounds just look really different from what your typical DEL compound collection would look like. Now, to screen them, we developed a workflow where we typically do this once we get up to about roughly on average 20 proteins of interest. We would tag them with a his tag and a streptavidin tag. We would load them onto magnetic dynabeads, then incubate with individual libraries or pooled libraries, where we hope that some molecules bind a hotspot here, maybe there's a second hotspot over here. A lot of compounds will bond non-specifically onto the beads, for example. We wash extensively, elute, PCR amplify, next-gen sequence, and now you've got a lot of data. So what do you do with that? Very noisy experiments, very challenging experiments. So for example, the barcodes themselves have unequal distributions. Well, Connor Coley, who was a visiting scientist, now is on the faculty at MIT, and Paul Clemens, who's been and remains the director of computational chemical biology at the Broad, got together and developed a data analysis pipeline. They're about ready to publish this. All of these instructions now will be freely available to the scientific community. And so we look for enrichment, but we also look for true enrichment by normalizing the barcodes. We look for structure-activity relationship, especially from stereochemistry. If we get enrichment always with one stereoisomer with different building blocks, but not with others, we're very comforted. And we would prioritize these then for synthesis off DNA. It is not a resynthesis, by the way. This is the first time the compound will be made to see if it binds. And of course, I wouldn't tell you this if it doesn't work. Even, you know, we knew it worked from the literature, but it works with these libraries we're synthesizing. Here's just one example. Shuang Liu and Andrew Reidenbach in the lab have been interested in the oncogenic allele of IDH1, R132H, and they screened that protein. And what they found was barcode enrichment for barcode 120, which told us this building, the identity of this building block, 37 that told us the identity of this skeleton, and 31 which told us the identity of this building block. Praveen Kokanda synthesized this compound right out of the screening collection, 43 nanomolar IC50. So it works. Now, I'm wrapping up. Where do we go from here? Well, what I said before is maybe these binders are molecular glues. That could be the case, but I think the field is transitioning in a really important way where we don't want to just discover molecular glues serendipitously. We need to pick the presenter and the target in advance. And for that, we need other kinds of assays. So we're sticking to our barcoded compounds. We think that's going to be a real advantage. We're looking at all kinds of ways, for example, to record a molecular glue event. That's a very exciting one, proximity-induced enzymatic activity. But we're also using affinity purification as we did in this experiment. And that's the more mature system. And so that's the one I'm going to finish this talk by sharing with you. Check my time here. Okay, I'll wrap up right here. So Jeremy Mason also joined the lab. As a Novartis postdoctoral scholar. He had trained with Bill Rausch as a synthetic organic chemist. And Jeremy wanted to pursue this idea. What if in building our library, we sprinkle in biasing elements for either a target or for a presenter? Then we could run affinity purification experiments. In the case that I'm going to show you, the library was constructed, a bespoke library, where the biasing element was added to favor binding to a presenter. So we have a library of compounds. Now what we do is we run one of those screens. We look for barcode enrichment. But now we add a stoichiometric mount of the presenter protein. So every single compound in the library in the second experiment is bound by its presenter. We know the KD, we use the concentration of the protein to make sure we're binding the presenter. And now we screen with the same protein. Now we can look differentially for barcode enrichment. We're hoping that this is a way to look for barcode enrichment due to cooperative binding of the presenter and the compound to the target. Now, I'm only going to show you the very first case. We did the normal thing. We tried to stack the deck to make it work. And we did that by picking a very well-studied system, probably everybody's first test case. We used the VHL presenter and the BRD4 protein as the target. And can we make this work? One of the nice features of doing this is if it does, you have a direct functional readout of successful discovery of a induced protein association because of the VHL adapter inducing ubiquitination of BRD4. Nothing novel going to come out of here. Lots of PROTAC molecules for BRD4 and VHL, but does it work? Does the system work? And it does. It works in this first system beautifully, and it's now been extended to a couple of other systems. So we're really quite excited about this new way to pick a presenter, pick a target, and find molecules that can pull them together for whatever purpose you want. In this case, as I said, it is indeed a degrader, very convenient for readout, but all kinds of other applications that I'll mention in a moment. So the barcode enrichment was night and day different in the presence of VHL. To test whether this compound really induces association, we use the nanobit system. We put the large bit on VHL, small bit on BRD4, concentration dependent addition of this compound shows in complementation of luciferase activity, stereospecifically, change the stereocenter, abolish it, take a linker or connector that seems to be about the same distance as this one, completely lose all activity. Now, colleagues at Novartis have recently solved the crystal structure of this compound. It's a very beautiful one. The compound, the connectors in a low energy conformation, it's making contact with both proteins simultaneously. There's lots of protein-protein contacts, there's cooperative interactions. And we are writing this work up and hopefully be able to present that formally in the future. And of course, as I mentioned, is there a functional consequence? Well, sure. This compound is a PROTAC. It induces the degradation of BRD4 in a concentration-dependent manner and a stereospecific manner, linker-dependent manner, as I described before. So in this avenue, what is my lab doing? We're making these bespoke CIP DELs, we call them chemical inducer proximity DELs, where we're biasing towards presenters, VHL, cereblon, but also FKBP family members and others, and biasing towards targets. I alluded to kinases earlier. We all know that there's hinge binding elements, there's conserved small biasing elements that can take molecules to kinases. The question is, can we find cooperative interactions with presenters to get to novel kinase inhibition? So where do we go from here? My final slide. I'll tell you what my lab is focusing on. So we're really excited about this very simple idea. Precision medicine, the right medicine for the right patient at the right time. What about in the right tissue? Could we make drugs only active in the organ, tissue, cell, or subcellular organelle of our choice? Well, a way to do that, like using CIP DELs, would be find the presenter protein that has tissue-restricted expression in the diseased tissue. Find a molecular glue that binds the presenter, where the molecular glue itself does nothing. The active agent is only the presenter small molecule complex, but that's only expressed in a limited way where we want the drug to have its activity. So there we generate then the therapeutic activity. So what kind of presenters could we use? Well, I mentioned the Vertex compound. You know what? FKBP12 with Rapamycin, all those other compounds with mTOR. Well, FKBP13 is restricted to the endoplasmic reticulum. That is where the Vertex drugs work. Now, you'd really like to get a presenter in the ionocyte, in the lung, in the endoplasmic reticulum. Maybe that's a tall order, but who knows? Here's another example we're excited about. When we found FKBP12, we quickly, after we found 13, FKBP25, 52, 59, we also found this curious protein, FKBP12.6. There aren't a lot of papers published on it, but those that have been published and people who study this protein know it's got a really unique tissue expression. That protein is expressed in the brain. GTEx shows the transcripts are in different brain cells within the brain, and proteomic databases reinforce this notion. So, you know, could we start making brain-restricted drugs by using FKBP12.6 as a presenter? So that's a little glimpse of where we're going. Let me finish with thanks. I tried to mention some of the trainees who've been working on this project together. Our critical collaborators, Paul Clemens, couldn't have done any of this without Paul. I'm very grateful to Connor and Paul's work on analyzing this data for us. The second part of the project is a collaboration with especially Fred Zicri and Corinne Briner. Corinne, many of you know, is the head of global chemistry at Novartis, and they have just been not only amazing collaborators, but really amazing mentors, and the trainees really have enjoyed interacting with them. And I mentioned Ben Cravat.